So, in this class of fundamentals of practical microbiology, we will be dealing with different types of culture, and now I will take you through some examples of cultures, different culture types that we will usually explore in our day-to-day laboratory experiments. Okay, so the video that I am showing here is completely all about different forms of cultures. First we will have a look at it, and then I will explain you individually so that you can very easily understand what are all these different culture forms. Okay, so these are all called as culture forms. So in this culture forms, if you see, some cultures are like liquid cultures. So like, let me say this particular culture that I am taking here is in liquid form, and also this particular culture that I am taking is in a liquid form, and this big culture that I am taking, this is also in liquid form. Whereas these particular culture that I am taking it are in form of plates. Correct. So these are all called as the Petri plate cultures. Anyhow, I will explain you the cultures very distinctly, but still this is not in a liquid form. These cultures are like in a solid form. It is semi-solid in nature. So you have some gel-like components which we term it as solidified medium for bacterial cultures. Okay. So and then we also have something like slant cultures. So this is what a slant culture is. You can see, I hope culture is there inside the slant. So this is like a solid based culture in which you use a test tube for culturing your bacterial organism. Okay, and also the same way you can culture it in test tube in a liquid form also. So this is a suspension liquid culture, and then this is, so now I will explain you what is the difference between a cultured medium and a non-cultured medium. If you see this particular image, this particular thing that I am showing you now, it is very clear that the left one, I hope this is visible. Okay. If you see, this left one is little more clear compared to the right one. That is what I said in the previous class, like turbidity. When the culture is grown, the liquid medium will become turbid. Whereas you see the left one, which doesn't have a culture, it is still in a normal, it's completely transparent. Now I guess you can very clearly see it. Correct? A completely grown medium and a completely non-grown medium. Correct? So this is the difference. So you can just by visualizing this, you can very easily understand how the cultures can be easily differentiated based on its appearance. Whether grow ayirka illaya, you can very clearly understand. Okay, now let me start how our cultures are being explored. If the cultures, how we are preparing, and I have also given you a Eppendorf tube in which I have something called as a glycerol stock. This is my mother stock. I have isolated an organism and I will store it in a particular protectant, a glycerol as a cryoprotectant, and I will protect it for further uses. So this is a stock mari enakku. So from this, whenever I require the culture, I will extract the organism from this particular glycerol stock. Okay, so if different types of conical flasks we are growing up, it is very simple. Just these are all scale ups. So this is 5 ml and a culture this I have to grow it in a test tube. Now I require some 20 ml you can grow it in some a little more bigger container. And then you can go for, if I require some 50 to 100 ml of medium, then I can go for something like a 250 ml conical flask. Now I need some 500 to 600 ml of culture medium. Then you can culture. Then you can go for 1 liter or half a liter conical flask for culturing. But still, if you require more amount of organism, let me say I require in something like 5 to 6 liters or 10 liters, then you have to depend on something called as the fermentor. I hope that fermentor and all, we are not going to touch upon fermenters, just the basic techniques. Okay, if you see, these are all, so this is a basic understanding of what is liquid medium is all about. Okay, now we will go into the solid mediums. So these are all solid medium. So this is a semi-solid medium. If you see, it is completely, it is not a liquid, it is there, it is like a jelly mar, it will be there inside. Okay, so this is called as an agar nutrient medium. So I have written it as, this is a nutrient agar, Na. So proper labeling is very important, then only I can identify it. So this I have prepared in 26/2, 2021. Okay, so this is a control medium. So you cannot see that, just only the medium is there. No growth of microorganism is seen in this plate. Okay, so if I inoculate the culture and if I allow it to grow, then this particular medium will have something called as the cultures. So I guess you can very clearly see the different colonies formed. So yesterday there was a question in your assessment, right? So people, I have asked you like whether can you visualize colony in liquid medium. How can you visualize colonies in liquid medium? Colonies can only be seen in solid or a semi-solid medium. Okay, in liquid medium you cannot just see these colonies, you can just see a suspension of cells. Take this as an example, you can just see the suspension of cells, not the colonies, exact colonies. You can see the colonies only in semi-solid medium. So these are all individual colonies. In this particular plate there are plenty of colonies, you can see, correct? I hope this is very clear, right? So these are all different colonies. So this is a sample of saliva. Just a 20 ml of your saliva, and a person's saliva has been taken and we have inoculated, and we have seen these many colony forming units are there. Okay, and then this is another particular streak technique. I hope this is visible. You can clearly see it, right? So this is better, I will show it in a white background so it will be a little more clear, I hope. Yes. Okay. Correct? So this is in a quadrant streak. So in this quadrant streak you have a pure culture. Okay, whereas in this particular plate that we actually took into consideration, here we had something called as different colonies. They are all not same bacteria. They might not be same bacteria. But here you will only have same bacteria. This type of quadrant streak that you are having is only for so I hope now it is visible. So this is a quadrant streak. You have 4 quadrants, 1, 2, 3 and then 4th quadrant. So this is a quadrant streak. Okay, I hope it is clear. So whereas now let me go to the another example of colony culture. So these are all colony plates. So this is a sample that is isolated from waste dumping area. Nama domestic waste, nama veetla garbage la collect pandrangalla, adu poi where we store it, and in that place we have collected the sample, soil sample, and then we have isolated. So these are all the different colonies that has formed. Okay, so this is another colony plate, and this is like a multiple colony plate. So these are all over streaks, the streak that I am showing here, and the streak that I am showing here, these streak that I am showing here, this one and this one, they are all individual colonies, but I have streaked in a single plate. Okay, so these are all different colonies. These are all different solid agar medium plates in which I have isolated my organism in different forms, like in colonies or in terms of multiple streaks or in terms of like quadrant streaks, correct? So this is how you have to very clearly understand about the handling of your cultures. Okay, basic handling, this is how the culture will be. So how my culture, culture look you can very clearly understand. And this epigraph, this is a glycerol stock. So this organism grow you need a source. That source is nothing but your glycerol stock. Okay, so this is all up to the basics of culture types.